Hi, thanks for tuning in to episode number 29 of the Doctor Podcast Show. I'm Dr. Robert Seichert, your host today. Today, we're going to be discussing a very important topic, and that topic is cancer. According to the latest CDC data, there are approximately 1.6 million new cases of cancer in the United States every year, which is unfortunate. And even more unfortunate, about 600,000 people a year die from cancer. In the past, we treated cancer with surgery, with chemotherapy, with radiation, and in recent years, we're treating it with immunotherapy. But there's a new development in the prevention and treatment of cancer, and that's cancer vaccines. So today we have a great guest, Dr. Irina Shkundina, and she is a senior research investigator at the University of Pennsylvania and at the Penn Institute for RNA Innovation. Dr. Shkundina also works with Dr. Drew Weissman, who's a world-famous researcher. He won the Nobel Prize in Medicine just recently in 2023 for research and innovation that led to the creation of the COVID or coronavirus vaccine, which we heard about in the last few years during the pandemic. Thanks very much for coming today, Irina. I appreciate your time, taking the time to discuss this topic, which you're an expert in. Tell us about your education and background and how you got involved in this type of research with cancer vaccinations.
Thank you so much, Robert, for this invitation and for this opportunity to speak here. So I come from Russia, where I received my undergraduate degree in biochemistry from Russian State Medical University, and then I went into a Ph.D. program in the laboratory of Michael Tarman's son, also in Russia. I've done some research in yeast prions, and there was a famous lab in yeast prion research. After completing my PhD, I worked in the lab of Konstantin Severinov on bacterial antibiotics. And at some point, I decided to move to the United States, where I actually started working in cancer in the laboratory of Alexander Mazin and continued in the laboratory of Chris Eischen. And just recently, less than 1 year ago, I've joined the lab of Dr. Drew Weissman to work on cancer vaccines. So we kind of decided to unite our efforts. Drew is a great expert in immunology, and I worked in cancer, so he offered me to work in cancer vaccines. And this is a new direction in his lab because the majority of people in our lab work in vaccines for viruses, against viruses. Sorry. Right. So this is a new direction, and I'm kind of the first one to start this cancer vaccine work. So we are only in the beginning of our journey, but that's a great start because it's a great lab and great supervisor.
Right. That's amazing that you're working with a Nobel Prize winner, Dr. Weissman. What's it like working with somebody who's that brilliant and innovative and is known worldwide for all his innovations? What's that like?
Yeah, so Drew Weissman is not only a great scientist, but just a great person to work with. He's very kind, very understanding, and the great thing, he appreciates his people a lot. So, and he allows people to speak their ideas and to implement their ideas. So that is, when you come to Drew and tell him this idea, he always tells you that this is a good idea, but then this is a good idea. So it's a good to hear. Even afterwards, he can say, it's a good idea, but you need to be careful with this and that. And I can start to realize that maybe not the great idea, but it's a good thing that he's encouraging people to think and to allowing them to work independently.
Right. I noticed you have a large team in your lab that you work together. This takes, I assume, a lot of teamwork and a lot of effort. with many people with various backgrounds in the field. Is that right?
Yeah, right.
So you're currently working, I know, on cancer vaccines, and I think there are other labs working on that as well. Tell us how cancer vaccines might work and what is the mechanism for their effect.
So the mechanism of cancer vaccine is that cancer vaccine trains immune system to recognize the cancer cell. So the cancer vaccines, they are specific for the cancer antigens. So the tumor belongs to the body, so it composed of the cells of the human body. So in that sense, immune system kind of sees it as its own, and they're not always wants to, attack. That's how the tumor grows, because immune system allows it to grow.
Right. Normally, the immune system is supposed to get of abnormal cells that don't belong there, but somehow tumor cells are able to evade the immune system. It tricks the immune system into thinking the tumor or cancer is okay, and then the cancer grows, right?
Yeah. So it develops like an immunosuppressive microenvironment around the tumor, and that allows cancer to grow.
Right. So how would a cancer vaccine work to either prevent or I should actually say, do cancer vaccines prevent cancer, or is it a way of treating cancer, or is it both?
It's actually both. So the majority of current vaccines that are already now in clinical trials, they are therapeutic vaccines. And they are either vaccines directed to a certain type of cancer, like melanoma, pancreatic cancer. There are also vaccines within the certain type of cancer. Companies now leading clinical trials to make patient-specific vaccines for cancer.
Patient specific. So you're basically
Yeah.
using the
So
patient's antigens to create the vaccine.
yeah, right. So the thing is that the tumor has a lot of genetic instability, and there are mutations in a tumor. So the how do we need to create a vaccine? We need to select this antigen in a tumor that is different from the whole body. So for several types of cancer, there has been some antigens found that can serve as an antigens for cancer vaccines. Also, the tumors in every patient are different. So what works for one patient with melanoma might not work for another patient with melanoma. So what now, like companies such as BioNTech and Moderna, they are leading the clinical trials with so-called neoantigen vaccines. When they take tumor from a patient, they sequence the tumor, the DNA of the tumor, they compare to the normal genome of the patient, to the normal, like blood cells, for example. And also using computer algorithms that allow them to select, because then when they just subtract, what is expressed in the tumor to what is expressed in the normal cells, they get a lot of candidates. But using some algorithms, they select the most candidates that will most probably work, like the most important candidates. And
Right.
then from them, they're making mRNA for those candidates. And they can use like 5 to 10 proteins in the same vaccine. So create a vaccine with like 10 different antigens, and then they immunize the patients with those vaccines. And then they do like, 10 maybe injections, and then they see in the end how the patient is progressing with. And they do it with the, with disease, which is, like melanoma, third or fourth stage, with the patients that, are failing other therapies, fortunately.
Right. So basically you're taking antigen from the tumor, are you reverse engineering that antigen, that protein from the tumor, and making messenger RNA from that antigen, and then the messenger RNA can create more antigen, which you inject into the body? Is that what you're doing?
Yeah, it's not what we are doing, because for neoantigen vaccines that are specific for particular patients, there needs to be a big facility to do that. We're just a basic science lab, so we're not doing neoantigen vaccines. We're not sequencing patients' genomes. We're just trying to find some antigens that we can work with, that will work for all patients with a particular cancer, or with a group of patients with a particular cancer. That's what we are trying to find. One of the difficult tests for cancer vaccine is to find an antigen to immunize with. And then there are other difficulties, like how to find like immunosuppressive, tumor environment, which is also another big challenge in cancer vaccines.
Right. Now, for the audience, you mentioned mRNA or messenger RNA. I think people are familiar with DNA by now, but can you explain the difference between mRNA or messenger RNA and DNA? What's the difference between those two things?
Yeah. So messenger RNA is like a middle guy between DNA and protein. So we all consist of proteins, and we have the genes to encode these proteins. And then the messenger RNA is a middle molecule that carries information from DNA to the proteins, and this is the messenger RNA. So if we inject messenger RNA into the cells will create a protein from this messenger RNA.
Right. So you can create large amounts of protein, and they can be customized and be patient specific, actually, right?
Right.
So the patient is less likely to have any side effects or adverse reactions since the protein is patient specific.
So the patient specific is not always the case. As I already mentioned, neoantigens vaccine, we are not working on neoantigens vaccine because this is what big companies do. But so mRNA, when it's injected into the body, is degraded like in 36 hours. So the protein is translated, is synthesized, and it has the time to train immune system, but then it's rapidly degraded. So, and that kind of allows less, toxic effects. It doesn't integrate into, mRNA does not integrate into the genome. So it allows protein to be expressed, and then it's gone.
Right. Got it. So tell us about LNP, or lipid nanoparticles. I believe this is a way to get the messenger RNA into the body and to go to specific areas. Can you explain that, the LNP or lipid nanoparticles?
Sure. So the lipid nanoparticle is like a vehicle to bring not only mRNAs, can also bring any therapeutics into the body. It's kind of protecting the molecule. And if we inject mRNA, it can be degraded very rapidly because there are a lot of RNases, and just RNA will be degraded and will not reach the target site.
I see.
So lipid nanoparticle protects mRNA first. Then it also different LNPs have different composition. And depending on their lipid composition and their charge, that they can go into different parts of the body. Some of them go to the liver, some of them go to the spleen, and some of them go to the lungs. And people do, a lot of work on creating the LNPs that target, different sides of the body.
Right. So this is like an envelope, the LNP almost that you stuff the messenger RNA into. The lipid nanoparticle protects the messenger RNA so that it can get to the part of the body that you're interested in and target a specific protein. And then the LNP kind of opens up and releases the messenger RNA when it gets to the address that it's supposed to go to. Is that how it works?
Yeah.
That's pretty fascinating. So you need different LMPs for different organs, and basically you have to customize that.
Yeah.
And you have to make sure they don't degrade on the way there, right?
Yeah.
Hey, you mentioned an interesting thing, and it's been mentioned by scientists and lay people as well. There's been some concern about the COVID vaccine specifically, that maybe the messenger RNA is producing DNA. It's actually going backwards instead of producing protein. It may be creating some new DNA from some enzymes that exist in the body, like it's called a reverse transcriptase enzyme. And there's some concern by some people that you may actually be introducing foreign DNA into the human cells by some mechanism where the messenger RNA encodes for DNA. Is that a reasonable thought or concern?
That doesn't seem like a reasonable thought, because human cells do not express reverse transcriptase. And so the only reverse transcriptase can be in the human cells that come from virus. But since viruses, reverse transcriptases are known to be specific to viral RNAs, so it seems like this concern is not, very relevant.
I think there were some in vitro studies where they found some DNA in hepatocytes or liver cells that seemed to be a product of the messenger RNA. So I don't know if that's been shown in vivo in a living person, but I think there were some laboratory studies that may have shown that. But you don't think that's a
Were these cells
reasonable?
infected with a virus, or
Yeah, I forget exactly how they introduced the messenger RNA into the in vitro hepatocytes or liver cells, but apparently they then found some DNA that had been transcribed from the messenger RNA. What if a person has some reverse transcriptase in their body from some other virus infection, for example, HIV, and somehow the two get together in an extremely rare event? Is it possible for that to occur? The other concern is some people are thinking, again, there's no definite proof of this, that the messenger RNA or the DNA product from the messenger RNA may introduce itself into germ cells, in other words, into sperm or eggs in the ovary, and then be passed down into subsequent generations. Is that science fiction, or is that a remote possibility?
So since the lifetime of mRNA in the body is not too long, I don't think it's the probability of it is significant probability of that to happen.
I see. So
because the mRNA was in the body, for years, and then for months even. But mRNA is rapidly degraded. So
right. Unless it's accidentally reverse transcribed into DNA, which again you mentioned is unlikely to happen. So yeah, we'll see what happens, but it seems unlikely. Are there currently cancer vaccines in clinical trials?
Yeah, there are a lot of vaccines in clinical trials. Companies such Moderna, BioNTech has a lot of cancer vaccines in clinical trials.
I see. So those are the companies that created the COVID vaccine, so they have
Yeah,
experience in it. Is that a messenger RNA based cancer vaccine or some other type of vaccine, if you know?
yes, there are a lot of messenger RNA type
Oh,
of vaccines that they are currently in clinical trials. There are vaccines, melanoma vaccine, pancreatic cancer. There are like solid tumors, neoantigen vaccine. So it's like when they, take away the solid tumor and sequence it, and then develop vaccine specifically for a particular patient that do that for many solid
right.
tumors.
So currently it's limited to certain types of cancers, but potentially if this works and we figure out how to do it, we could target just about any cancer because every cancer has its own antigens or proteins that could be targeted. Is that right?
Yeah, so but potentially could work for every cancer, but there are challenges such as finding the antigens, specific antigens. So the vaccine will only target cancer cells. Also, there is such a problem as tumor heterogeneity because tumor may have several clones that are different from each other, and even when they take cells from the patient from one, even they take from several parts of the tumor and create the vaccine, there may be another part of the tumor that will give new like sprout for new cancer.
Right.
This can fight like some of the part of the tumor, but another part of the tumor will grow. So this is another challenge. And the third one is immunosuppressive microenvironment, because the tumor is surrounded by this microenvironment, and then the cells of immune system just cannot get to the tumor. So a mechanism preventing to getting cells to the tumor.
Right. Just a question. The tumor contains this antigen that you're trying to attack. Why isn't the body attacking the antigen on the tumor enough, or why isn't the immune system attacking it? And is the purpose of the mRNA vaccine to just create huge amounts of this antigen or protein so that the immune system is put on alert because there's so much of this protein floating around? Is that how this works?
Um, so not exactly. The purpose of the vaccine is kind of to reintroduce this antigen to the immune system and to train immune system to recognize this antigen. That's how it works.
See, why doesn't the immune system recognize it when it's on the tumor? Why do you have to
Yes.
send in the extra mRNA and extra antigen? Is that known?
Yeah, so there are some antigens that have low level of expression.
I see.
So immune system doesn't see them or the presentation of the antigens. There are also the molecule that present the antigen to the immune system might be also some low expression of those molecules. Also, tumor suppressive microenvironment where the tumor cells express the molecules that interact with immune cells. And they tell them, okay, we're not foreigners, we are okay, and tumor cells not attack them. So, and there is an immunotherapy, which is called immune checkpoint inhibitors, where
Right.
they block these interactions.
Right. Some of my patients who have melanoma are on those medications. They've been very effective for many patients who've survived a long time and wouldn't have
Oh,
survived without them. So
yeah.
we're making good progress. What about using mRNA to as a vaccine for treatment for certain types of diseases, not cancer, for example, malaria or tuberculosis or other types of infectious diseases that we don't have great treatments for? the messenger RNA vaccines be used for that as well?
Yeah, for sure. So there are vaccines um, for HIV, for influenza, and I think, yeah, malaria and tuberculosis as well in clinical trials right now.
Oh, so
And
there are trials right now. That's great.
yeah, and also our lab is also working on influenza and HIV vaccine. So we're hoping to get something
That
at some point.
that would be great because HIV for decades we still haven't been able to come up with a vaccine for it. So maybe this new technology with messenger RNA may do the trick. What about CRISPR has been in the news a lot. It's a basically allows you to edit DNA. Do you use CRISPR also to edit the messenger RNA, or is that a different way that you're doing that?
No, we're not using CRISPR to edit messenger RNA. So CRISPR, it's on its own can be a therapeutic for, let's say, genetic diseases when the CRISPR system deliver to particular. I'm not an expert on that, so
Yeah,
CRISPR can be
it's been
therapeutic.
recently approved for treating sickle cell disease and also thalassemia, which are two blood diseases. So
Yeah. So
it's been
for genetic diseases, different platforms, different CRISPR platforms are being elaborated for that, and also the companies work on that. But with RNA, we're not doing anything with CRISPR.
without getting into proprietary things, how do you modify or edit messenger RNA if you're not using CRISPR?
So
Or-
the modifications, Dr. Karika talked about it in her Nobel lecture. So this is
Oh, okay. Yeah,
this is not,
she worked with Dr. Weissman on the COVID vaccines, right?
yeah. And so the modification, so mRNA consists of 4 nucleotides, and one of the nucleotides, such as uridine, is substituted for pseudouridine, which has the same chemical composition, which is an isomer of uridine. And when they found that this pseudouridine, when it gets incorporated into mRNA, it ensures better mRNA stability, and also this modified mRNA does not cause inflammation.
So the
So
body tolerates it
yeah,
and doesn't destroy it right away. So
for
yeah,
mRNA itself, yes, and doesn't destroy it. Yeah.
well, that's a great discovery, definitely worth a Nobel Prize in medicine. When do you think we'll see the first cancer vaccine available for treatment after going through the clinical trials? Based on your knowledge and experience, when do you think that might happen? I know it's difficult to predict because they have to go through the trials.
Yeah, right. So the vaccines for melanoma and for pancreatic cancer, they showing like from 35 to 50% efficacy in clinical trials, which is considered to be good. So it might be, I don't know, but might be
Right.
new vaccine will be around soon.
Right, hopefully.
Uh, yeah.
Another question that when we use the word vaccine, we typically think of it as a way to prevent diseases. For example, measles vaccine or polio vaccine is given to prevent those infections. Do you think we're eventually going to have cancer vaccines to prevent certain cancers? For example, we know that people who smoke cigarettes are extremely high risk for developing lung cancer. Will we get to a point where we have a lung cancer vaccine and anybody who's been smoking just gets that vaccine and it prevents lung cancer? Is that a possibility down the road?
Yeah, it's certainly a possibility, and we're also working on preventive vaccines in collaboration with other scientists. And there are such cancers that hereditary cancers. So patients with BRCA mutations, they have predisposition for breast cancer.
Right.
And so these patients are, if they have like a family history of breast cancer, sometimes they are dependent on their family history. They have to just get their, breast removed or ovaries taken out,
Right.
before they when they reach 40 years, which is very traumatic and in many senses. So it would be much better for these patients to get a vaccine that will, clear out any first signs of cancer. And
are there any preventive mRNA cancer vaccines in clinical trials currently, or is it just the treatment vaccines that are in trials?
I think there are also some trials on the use of cytokines for cancer. I'm not sure, are they at the stage of the trials, or it's I think there is there are like first stages of clinical trials in using of cytokines, which modulate immune system, and they can stimulate immune system to attack cancer cells. And that's what
That would
we're
be
hoping for as well.
that would be an incredible accomplishment, because many of my patients, for example, have various genetic diseases that predispose them to cancer. So they're always being tested, and they're always nervous and worried about that. So that would be an incredible accomplishment. Well, I'd like to thank you very much for taking the time to join us today on the Doctor Podcast Show. This was extremely informative and educational for me, and I'm sure the audience as well. And I want to congratulate you and Dr. Weissman for the incredible work you're doing, which is helping millions and billions of people around the planet. So thank you very much for coming today, and say hello to Dr. Weissman for me as well.
Thank you so much, Robert, for inviting me here. I also like to thank Dr. Drew Weissman for giving me such a chance to speak with you here. It's been a pleasure.
It's been a pleasure. Thank you very much, and have a good evening. Bye-bye.
You as well. Bye.